afternoon, ladies and gentlemen, and thank you for joining us today, today's Live from the Lab seminar. That beautiful piano piece you heard was actually performed by our own professor, Matthias van Herrath, the star researcher whose work on diabetes you will learn about today. I am Erica Ollmann Saphire, and I have the honor of serving as this institute's president and CEO, as well as being a research professor here as well. This institute is a special place. It is a nonprofit think tank where our goal is to use the tools and the minds of the visionaries that work in this building to imagine a path forward or a life without the diseases that just happen to our families, whether they be type 1 diabetes, the subject of today's seminar, or cancer, or autoimmunity, or infectious disease that compromised so many of us for the last couple of years. But we don't get up in the morning just to imagine things. We go to the lab every day to make them come true, to bring about a time when we don't just suffer from these things, but we can understand the mechanisms by which diseases emerge and find the ways to detect it earlier and treat it and erase it so that other families don't suffer the way we have. We are an independent, nonprofit biomedical research institute. What that means is that we are a freestanding think tank and a 501c3. We're a tax-exempt charitable organization. All of our resources go to research to make new treatments to improve human health. And more importantly, because we are a freestanding, nimble, nonprofit think tank, we're an organization run by scientists, for scientists, for science. Our mission is this: that the Institute will engage in a world-class biomedical research program with a focus on the immune system. And we focus on the immune system because of its incredible power and complexity, its ability to find and destroy individual cancer cells that no scalpel can find, its ability to slow advance of heart disease, the ability to defend against viruses we have seen and ones we have yet to uncover. The immune system plays a central controlling and sometimes causative role in every human disease. So what we can understand about the human immune system will help us in every aspect of human health. Our institute conducts, shares, and partners such that the results of our discovery program, and this is important, will make outsized contributions to the betterment of human health. This small, focused institute very much punches above its weight. We're 34 years old and we have 2 independent labs, each with a different focus and each with different tools. But this small group ranks 5th in the world in scientific impact in the area of understanding immunology, and human health. We have about 500 people that work and propel our research, including over 170 technicians and support staff, and 200 young postdoctoral fellows and other trainees that launch their independent research careers from here. So we have very much an expansive catapult training role as well as the fundamental research that happens in our own building. Our 22 faculty are loosely divided into 3 areas. There are some of us that study infectious disease and vaccines. There are others that study autoimmunity and inflammation, and there are more that study cancer. When I say loosely, it's because some of the tools we use very much can focus in different areas, and the kinds of immune cells that we study may play controlling roles in multiple different diseases. And in fact, that's one of the beauties of LJI, is the cross-fertilization and cross-pollination of ideas. Today's researcher is my good friend, Dr. Matthias van Herrath. Matthias joined LJI in 2001, and he came for that interactive collegial environment. He did his medical degree in the University of Freiburg, studying medicine for the opportunity to make a difference for human health. And that's what he came to LJI to do, to discover things that hadn't been put into clinical practice yet, so the doctors would have more tools at their disposal. And in 2012, he did something very creative. He expanded beyond the research lab to partner with and work closely with Novo Nordisk to make sure that what was discovered in the laboratory was brought to clinic for human health. I'm very excited to see his new research today and understanding the mechanisms by which type 1 diabetes manifests and what we can learn to slow and reverse disease. Matthias?
Erica, I'm so happy to be here, and thank you for this very nice introduction. And I'm excited to tell you today more about type 1 diabetes and some moonshots, maybe how diseases such as type 1 diabetes could evolve. And also, I would like to update you where we are in terms of translation and treatment. Now I have to find my PowerPoint file, and then I can probably present things. Gina, help me. I'm on an Apple computer. I'm not usually on an Apple computer. We have to go there. Ah, there we are. Excellent. See, I knew you had to help me with this. There we are. Good. Yes. So we need better insight for diseases such as type 1 diabetes, probably in a broader sense for other autoimmune diseases, in basic research, because these diseases that are likely caused by multiple factors, and that's important to understand. That means genes, environmental factors, are difficult to unravel and to find the primary cause. And
Dr. von Herrath?
Yeah.
Sorry to interrupt. Would you mind just showing your slides one more time?
Okay.
Perfect. Thank you.
So, and because of that situation, it is our belief, and this is the work, what we're doing here at La Jolla Institute, we need to better understand the human pathology of diseases. And we will have some basic considerations about type 1 diabetes. And just to put it out front, and we will stay with that theme, understanding pathology of human disease can be quite insightful. It's like the game Clue. And just in the game Clue, you try to understand who the culprit is with which murder weapon at which location. And that's really what we are trying to do here, as to then infer how the human diseases are caused, and based on this to understand how to treatment. It's very important to understand how complex these diseases are. It's not only type 1 diabetes, it's alopecia, it's rheumatoid arthritis, all these diseases where we seemingly think that our own body is being attacked. And we think also now at this point that in addition to the immune system that definitely plays a role, other factors will contribute to this. And the concept I want to introduce to you today is that the targeted organ in diabetes, the beta cells that make insulin, probably contributes to its own demise. And maybe in this case, you will hear about this through innervation. So it's sort of an unveiling of your own tissues to the immune system, which is of course not a good thing. And we think that factor, rather than just the immune system being inflamed or overreacting, that tissue factor, that wrongdoing of the tissue, its unveiling to the immune system is a very important factor to contribute to the disease. And it underlies what I call a moonshot in this concept. The moonshot would be, how could we find something like traveling to the moon that would alleviate many diseases, not only type 1 diabetes. What could be a common pathological theme of this unveiling of the targeted organ to the immune system? These autoimmune diseases are a huge problem altogether. There's many chronic conditions. There are millions of people affected, and some have more than one autoimmune condition. But it again is important to understand that many of them are quite specific, which puts also more emphasis to this unveiling of a specific organ to the immune system. And I start at something that we are kind of all aware of, but it's really, really unsolved. It's an unsolved enigma. Autoimmunity and its manifestation. And you see that's also true for the pancreas. And it's certainly true for diseases that you can see well. It's often spotty, but not only that, it's often even symmetric. And it comes back to what I just said. If it's symmetric, if it were just an immune overshooting or a hyperactivated inflammatory immune system, why would it be symmetric? Why would it be only in certain locations? Why wouldn't it be in the whole body, the whole skin, all the eyelids in the pancreas at the same time, all joints in arthritis? But that's not how it is. You can see this here. For example, in lupus, there is a certain rash that comes with the disease. Lupus is a systemic, severe autoimmune disease with heart and kidney participation, difficult disease for the patients. And it has this rash. And here you already see it's called butterfly rash because it's just around the face. And we can ask, why there? Why not somewhere else? Why not on the back? Why not on your feet? So these are very unsolved enigmas. It gets more impressive when you look at arthritis. Also there you have this symmetry, but it's only certain joints at certain fingers. You can see here, you get certain joint affectations in the fingers, but it's only certain joints. It's not other joints. It's also the same for the feet. And maybe most impressively, another autoimmune disease is the disease vitiligo, where you attack the cells that produce the dark color in the skin. So then the skin becomes white. And you can see here in this case, it's highly symmetrical. And again, the question, why would it be just certain areas? And why would it even be symmetrically here in this case to the T? You can see on the ring finger, it's even just on certain areas. You can't just explain this with sun exposure and so forth. So there has to be something else. And that we will come back to. And as I mentioned in the beginning, we think that innervation of the target organ in autoimmunity is very important for that, and probably an entirely new concept of how autoimmunity could evolve. When you come to the pancreas, same situation. In the pancreas, you have areas as demarcated here. It's maybe not as impressive to see because we're now starting to get into the clue game of who is killing whom in the organ. But you see areas, red is for alpha cells that make glucagon, green is for beta cells that still make insulin. There's areas, the beta cells, the green staining is still there. Then there's areas where the eyelids are gone altogether. And then there's areas where only alpha cell or red eyelids are there that have already lost the insulin producing beta cells. So it's a very spotty disease. And again, you can come back to the question, why is it only certain lobes of the pancreas? Why not the whole thing if it's a systemic overreaction? And so this organ specific contribution to its own demise or undoing remains a very, very important concept, but not very well understood. People say, well, it's viruses, but how would a virus just infect, like, for example, in a rash, a certain area symmetrically on your skin? It is not conceivable. And the relationship with viruses and diabetes, it's maybe there. There is more virus infections in general in the diabetics pancreas, but a cause that one virus would cause this disease or set off the process that then manifests itself as type 1 diabetes has never been established. And there's over 30 years of research. So viruses might be accelerators and contributors, but probably not in most cases the primary cause. And they are often, when you see something here on the right-hand side with shingles, indeed, there can be a localized situation. In this case, a virus gets reactivated from nerves and causes a rash, very painful one. But it's not symmetrically. So it is not such an easy culprit. And in type 1 diabetes, we have worked on this for the past many years, decades actually. The unveiling is quite interesting to see. This is an eyelid where the green are the cells that make the insulin that we all need to keep our glucose in check and store it actually in the organs for use. And red is the whole eyelids, which also contains other cells. And you can see this red staining is an immune recognition molecule, and that's upregulated in type 1 diabetes. Almost like the eyelid would say, well, here, over here, if you can see me, now you can recognize me and you can come and attack me. So it's like an unveiling, like I said, in the beginning of my talk. And it's indeed so. This is a movie we made in the mouse. When you have these little green cells, the little green dots are your attacking cells. They're swarming around the pancreas, sort of like anthrax in the kitchen. In California, we get that very often. And then the green blobs, the big blobs, are the eyelids. And that's like sort of the food you have left the evening before on the floor accidentally for the ants. And the ants, as long as the food is revealed, and they probably have all kinds of pheromone receptors for sugary things, they will find the food. We all know this. And that's somehow how this works in the pancreas. As soon as the islets send a signal that they're here, then the scouts of the immune system will find these islets and they will begin their destructive path. And it looks very similar actually to anthrax. That's why I like the movie. So in this organ surveillance, that we have cells that react to certain components of our own body and they come to survey the organs, that is actually happening notoriously everywhere. Otherwise, we wouldn't find virus infections. So we have these scouts at all the organs and we need them as an immune surveillance crew, and certainly to keep viruses at bay. And here something goes wrong because they start recognizing our own tissue pieces. So to summarize this, and a very important concept, the islets are somehow instigated to reveal themselves to the immune system. And we think, and that's my sort of out-of-the-box moonshot thinking, we think the innervation might play a role, not only in type 1 diabetes, but other organ-specific diseases. And actually in alopecia, you also see what I show here on the left-hand side, that these recognition molecules are upregulated. And then the little cells, they are here, insulin is green in this case, they are red on the right-hand side. They come and recognize these beta cells that have now given the signal to reveal themselves to the immune system. And we just recently published this in Science Advances, that the cells that can actually find islets, they're already hanging out in the pancreas and they are recognized pieces of insulin. So just to drive this home one more time, there's a healthy beta cells. Our scout cells are not very interested in them, even if they might be able to recognize pieces of insulin presented to them. Nothing is revealed. The recognition molecules are not there. They survey the organs and they look at these healthy beta cells and they remain uninterested. They might even really check them out. And then if the beta cells reveal themselves, these prowling cells really come to the attack. Now, I will turn things over now to Estefania Quesada-Masachs, who has been in the lab for over 3 years. She is really the one who manages many things, and she's the one who is usually at the crime scene, coming back to the clue analogy, and she does the crime scene investigations, and you can have a real life view right now on these crime scene investigations in our wonderful microscopy core. All to you, Estefania.
Good morning. I'm Estefania Quesada-Masachs. I'm one of the postdocs at Matthias von Herrath lab. And as he said, I am in the microscopy core. And the reason is because we have a lot of collaborations with our microscopy colleagues, and also because we use a lot of microscopy in our studies. And you may notice that this room is actually very dark. And the reason is because we have the confocal microscope here. Confocal microscopes work with lasers, and lasers are light sensitive, and this is the reason why they need this darkness. And we use confocal microscopes because they allow us to get what we call high resolution images. High resolution images means that we are able to see where different proteins of interest, which means some of these like guilty guys in the process of type 1 diabetes, we can see where they are expressed, which cells express them. And inside the cell, we can see precisely where they are located. in our lab, we work with human samples. We investigate the pancreas and eyelid structures that originate from human donors. And I'm going to show you some of the representative images that we can get that we actually got in some of our recent studies. So in the first image, I'm actually showing a small piece of a healthy human pancreas. And in green, you can see the insulin. And you can see that there are also some cells expressing white. This is CD68 that recognizes macrophages, and some cells that express red. If I show you all the images separately, this red is HLA class 2, that is one inflammatory marker. And actually, there is not much here because not many of the macrophages that you can see here in white are activated, and there is not a lot of inflammation going on in this pancreas because this pancreas, this is the healthy islet showing insulin, is healthy. Now I'm going to show you one pancreas of a patient with type 1 diabetes, and you can see the striking difference. Here in green, we also see the islets, but when I show you because this is recognizing insulin, but when I show you the images separately, you can see how there is not much insulin here. There is a bit less than in the other case. Also, you can see much more white, which means many more macrophages infiltrating the that are these CD68 positive cells infiltrating this islet. And you can see a lot of inflammation, a lot of red is going on. This inflammation, this HLA class 2 in this case, is expressed in the macrophages, but is also expressed in the insulin producing beta cells. So you can see when we compare these images with the images that I showed you before, there is so much more inflammation going on in diabetes, and these islets are clearly under attack. So also in the lab, we work with what we call in vitro systems. And in the in vitro systems, what we have are, for instance, pancreatic slices that are human alive slices that we can modify, and we can recreate some of the processes that happens in type 1 diabetes. And we also work with in vitro systems with human islets. And we have two different platforms that I'm going to show you here with the images. In the first platform, you can see the perfect islets, that these are called spheroids. These are human islets. The cells come from human donors, but these islets are perfect, and they are produced by the biotech company Inspero at Switzerland. And here in blue, you can see the images of the nucleus of the cells. In green, you can see the insulin again. So the islets have a high concentration. These are healthy islets, happy islets, have high concentration of insulin positive cells. This in orange shows glucagon, that are the alpha cells. And here in red, I'm showing HLA class 1, that is one molecule that can be present in all of the cells, all of the nucleated cells in the body. But when there is inflammation, the expression is much more upregulated, is much higher. Sorry for that. Because, somebody connected to the, well, things that happen when you are alive, right? When you are alive. So these are one of the perfect islets from Inspero. And we also work with the isolated human islets. And the isolated human islets, we get those islets from City of Hope. And these are the islets that are used in transplantation. And like our human bodies, they are much messier, right? Because some of them, they are larger, some of them, they are smaller. And if I show you the split image, you still can see that the same markers are expressed there. So in blue, we can see the nucleus. In green, we can see the insulin. In orange, we can see the glucagon to identify the alpha cells. And in red, we can see the HLA class 1 that can be, that is present in all the nucleated cells in the body, but that can be upregulated when there is inflammation. So with these in vitro systems, what we do is we can keep them happy or we can keep them unhappy. We can modify them, trying to recreate all of these issues that happen in type 1 diabetes. And I'm going to show you in this last image, again, isolated human islets. These are the natural islets, not the perfect ones. These are the ones that are used for transplantation, but these islets were unhappy. I stimulated them with inflammation, with inflammatory cytokines, and you can see how they express much more red, which means much more HLA class 1, which is one of the things that we observe in type 1 diabetes. And they express still insulin, but a bit less than the islets that I showed you before. Overall, the reason why we work with all of these human islets and human islet spheroids that we get from Inspero is because we want to recreate the processes that happen in type 1 diabetes. We want to understand what makes the islets happy, what makes the islet unhappy, and ultimately we want to modify and try to trick the process, because it's how we are going to move forward in finding better treatment strategies and potentially a cure. So I hope you like the presentation. If any of you is interested in coming, please let us know, because you are invited. And I hand the presentation back to Matthias. Thank you.
So thank you, Estefania. That was wonderful. And it's probably really the most important thing, because you were just a witness to the real investigation, where we investigate the crime scene before some more theoretical excursions and updates for me. So coming back to the topic, why human islets and other target organs might become sensitized to destruction or revealed to the immune system, I want to dwell a little bit more on a potential of the innervation of the target organ for causing these type of recognition events. The consequence would, of course, of this, what I call the big idea, would be that you could then modulate, we get into how we could do this in a minute, you could modulate nerve activity to prevent immune attacks in certain regions of our own bodies. And I think that could be very helpful if it turns out to be true. And it's an under-tapped area. It's an unresearched area, really, probably for right reasons, because it is not easy to envision how to modulate the nervous system. But I come back to that too, and one update to this. The first clue really we got, and this was studies in mouse models as a proof of principle, and we have already done those several years ago, otherwise I wouldn't bring you this idea, because we wanted to know, is this even viable? What happens if you cut the nerves to the pancreas? What happens in a mouse that has an attack on the eyelids when you interfere with medicines to switch off certain nerval activities? And what happens is that you can completely stop diabetes, even though you have a target organ and you have a cell that recognizes the target organ. So it seemed, at least in a model organism, the nervous system is able to tune and override all this. And that made us encouraged. We thought, well, if this can in principle happen, the nervous system is possibly quite important. Now, that doesn't make things easier. There's various systems of the nervous system, the parasympathetic, the sympathetic, and many other arms, and it regulates organ function. So you get it based on this insight. It's not so surprising that the nervous system could influence many processes. I still think it remains surprising that an innervation or lack of innervation can completely make an ongoing immune attack to subside and change how the organ is seen by the immune system. I find that actually quite intriguing as a potential therapeutic avenue. But you also see, when you look at what all the things that the nervous system does, we have to be extremely selective. There's no such thing as taking a systemic drug or inhibitor without side effects. And obviously, all the neuronally effective drug that we are taking, also neuropsychiatric drugs, they have side effects. And we have to become more selective altogether. And it is really not entirely clear how many diseases you could modulate and how many processes you could modulate. And we propose to do several things if I had my dreams come true, which would be nice sometime in our lifetimes to investigate this. We can now have these living tissue slices that Matthias in our lab is working on, and that Estefania has referred to already. And there you can modulate living human tissue that you can obtain from patients who had to undergo surgeries for other reasons or organ donors. And then even more interesting, there's now several device companies who have thought about this too and said, well, we need to modulate nerves very selectively. And these device companies actually are working on devices so you selectively can only modulate nerves at the site where it matters. And there's the company Galvani, who is thinking about transcutaneous electrical devices, also, for example, to treat pain. As a lot of these activities by these companies is driven by pain management. And the CEO of Galvani, a gentleman named Chris Pham, was elected in Time magazine several years ago as the most out-of-the-box thinking scientific individual. It's a small company in the UK. They're very interesting. There's, for example, Metavention. There's other companies who do this. I don't want to be selected. Metavention is, it inserts devices in vessels and can then disrupt the innervation through the vessel. So in principle, we have the basic observation. We have certain tools. It's probably a good time at this point to put this case together. Also because the implications, if we miss something here, the implications are vast. This could really change things quite profoundly, how we go about treating organ-specific, autoimmune, immune inflammatory diseases. And you can do various things. You could have skin tissue, you can have pancreas organoids, you can have these tissue slices, and then you could sort out which nerves actually need to be modulated to stop the immune system from attacking these tissues. And you could probably parallel start this, not only in the pancreas, but also in more accessible organs like the skin, where there are many autoimmune diseases, and then hope to treat the disease altogether. It would be local. It could be reversible. Technology-based is always cool because it gives the user some influence. You don't take a pill and then everything is said and done after they swallow the pill. Would, because of that, appealing to patients. You could control things, just like in diabetes, you have more control when you have glucose monitors and insulin pumps. And the meshing of biology and technology has been good. You can see this also in the involvement of the diabetes field. We have glucose sensors now, we have pumps, we have closed loops with pump sensor algorithm that in a way almost provide an artificial pancreas. So technology advancement has been good, and it seems sensible to marry that, because these things, like our phones, they have advanced much faster than understanding complex biology, and marrying them seems to be a sensible situation. Now, in the last few minutes, I just want to give you an update other than this innovation moonshots and the basic pathology and crime investigations in type 1 diabetes. But what we have done to translate things to the clinic, and it will relate to my other role where I work with Novo Nordisk for the purpose of bringing promising interventions to patients. The way the loss of beta cells is in type 1 diabetes is that you have this pre-diabetic phase where you still have a lot of beta cells. And if you prevent things there, you can expect to retain full organ functions. And Prevention Bio, with their anti-CD3 antibody, has recently shown that they can delay disease up to 2-3 years if given in the pre-diabetic phase. But even after onset, you still have beta cells around, and it's probably quite worthwhile to preserve them as measured by C-peptide in the blood of patients, and help patients with easier management of their glycemia and also independent by having C-peptide stave off complications. Last but not least, you could imagine putting stem cell derived beta cells that make islets, which is now possible, back into patients if you have the proper immune modulation. What are the obstacles? In prevention, certainly the obstacle is to find the patients and have screening, because it goes hand in hand with the drug offering. You screen a patient, you want to offer also to the patient a means based on the screening to then do something about the situation. In stem cell transplant, and some of you might have followed this, ViaCyte very active, Vertex very active, we can have these stem cell derived islets, but when they go in patients and they're not protected, you need full immunosuppression to make them functionally, not only to curb any recurring autoimmunity, but also to curb any organ rejection, allo rejection that you would see usually in organ in that situation. And we need the trials, of course, to sort this out. Where are we with the trials? We have made an antigenic tolerance that you could consider as an immune maintenance therapy to re-educate the immune system not to attack the insulin producing beta cells and recognize insulin. And that's in a phase 1 right now. And we have conducted the largest combination study where we have a component working for the beta cells at GLP-1 and an anti-interleukin 21 in adults with type 1 diabetes that was just published last year. And both of these discoveries, that's important to understand, they were made in the lab. So the basis of combination, induction, maintenance, doing something for the beta cell as well as for the immune system, as well as the concept of antigenic tolerance, which means specifically making cells of the immune system that recognize beta cells become tolerant to that. They were all developed in the lab over many, many years and decades. So that's how the system works. You need the basic research to then bring this to the fruition and develop this in context of a pharma company to make drug treatments for patients. Similar, this will be in eyelid transplantation. I'm sure many of you are interested in this. In my mind, the way this field will evolve, it's a triad. You have to have somewhat immune evasive beta cells that you make from stem cells. It doesn't have to be perfect, could be semi-perfect. You could see whether you have a capsule, so you keep them together. Also doesn't have to be perfect. It's difficult to make. And you have to have some immunotherapy that is tolerable for patients. That has to be pretty good. It cannot be something that is a profound chronic immune suppression. And somewhere in this triangle, there will be the sweet spot. And right now, why this is exciting is the time to actually find this type of a sweet spot in organ replacement for those who already have type 1 diabetes further into the disease. Timelines, what will the future bring? I'm often asked this, so I put this in, difficult to predict this thing. I think stem cells will evolve over the next decades to become a palatable therapy for patients, just like I said, in that triad of capsules, immune evasion, and mild immune modulatory therapy, as several companies are pushing the boundaries on that now. I think we have good combination therapies and recent onset. There is some regulatory hurdles. I would hope that regulators could eventually see that we really would need a therapy in the space that can preserve C-peptide as the main outcome and reduce insulin dose like we have shown. Of course, we would need better insulins. That is ongoing. Probably at some point, we at least will have a basal glucose sensitive insulin. And then in prevention, I already mentioned that we will at least have anti-CD3, which you can build on as an induction drug and then have maintenance therapies. So that is altogether actually a quite promising outlook in the field of type 1 diabetes altogether, because it's now an exciting time where many things are coming together and converge. Also in the clinical space, bearing the fruit of probably over 30 years of research and investment into this area. Where are the gaps? Research enterprise is difficult in terms of the competitiveness, the time spent on grant writing, which is becoming ever more. And we need to retain the best people and keep them interested in the field. Okay, that is a gap. It is a fundamental gap. We're not nimble enough in reviewing things and bringing things to at least a stage where we know we could build on that. So it will always be a gap to explore creative ideas. And we need cutting edge technology. Coming back to the crime scene investigation. I mean, everybody can understand that having DNA evidence has revolutionized how we look at murder investigation and the like. And it's not so different in science. If we have the worst equipment, you're not going to see anything, and you're definitely not going to push the boundaries. And you need the best equipment, and we're very fortunate here, but you also need to maintain the best equipment, and you have to ever evolve it. And coming with these examples of the iPhones, we all know that we have to about every 5 minutes buy a new iPhone to stay at the cutting edge of having them functional. I'm going to close there. And hopefully that some of you will dream together with me about some of the moonshots I presented to you. And thank you very much for listening in. I think back to you, Erica.
Thank you so much, Matthias. You know, we're all very fortunate to have the opportunity to spend our lunch hour with Matthias, as he is one of the world's experts in understanding type 1 diabetes. And those of us in the building who have the honor to work with them are even more privileged. Thank you for bringing up the instruments. You know, you can do the best research if you can see at a level that hasn't been seen before. And it was exciting to be able to go into the microscopy core and see those live cells, because seeing those interactions is really what leads to discovery. And that's what leads to understanding the mechanism of putting a treatment in the hands where there hasn't been one before. So we have some questions from the audience, and I have the first one here. So Matthias, this is a wonderful one. Let's talk about viruses. Let's talk about viral triggers of autoimmunity, and if there's a smoking gun or no smoking gun. And more interestingly, how about endogenous retroviruses? Is there any opportunity to use that information? Can you treat type 1 diabetes with an antiretroviral drug?
Yeah, so this as I mentioned in my talk, that has been around for a long time, where we converge with our evidence. And I started with Michael Oldstone, who was a virologist at Scripps in 1991. It seems like awfully long ago in these good old days. Where it converges is that probably viruses, maybe even endogenous retroviruses, could be accelerators. It is true, for example, that you find more enteroviruses in recently diagnosed patients. But it has been difficult to identify one culprit. And the epidemiology of virus infection and where they are found doesn't support that there is a single culprit causing type 1 diabetes from the get-go. And that's where the field is. That doesn't make them less important. If they're an accelerator, they're an accelerator. We should do something about it. And friends in Oslo are running an antiviral trial right now. I think we will hear about this right now this year. And it will be interesting to see how much of an accelerator of diabetes they can be, and whether, maybe even with a viral vaccine, whether we can improve things altogether. And to that notion, we probably see some of that with COVID, this accelerating factor in these autoimmune diseases.
Indeed. Well, here's another question. So the immune attack on the eyelids, if we have a pancreatic transplant, an eyelid transplant, are they also susceptible to the same type 1 diabetes that destroyed the original eyelids? Is this linked to innervation? Does innervation cause it? And then a second question from the same audience member, the role of antigen presenting cells, like dendritic cells, in the initial immune attack on the beta cells.
Yes, I take them one by one, and actually the second one first. Estefania is actively working on this. And we have just completed a study that I'm quite excited about, where we looked at macrophages and these type of cells in the pancreas and their proximity to eyelids, sort of really like this clue investigation. How close are they? When are they close? Might they have something to do with this? It's sort of like this murder scene thing, whether you were there or not there, and did you have any weapons with you while you were at the scene? So I think stay tuned for that. That's looking pretty interesting. That's the second question. The innovation question is more complex in the context of transplantation, and it is a very good question. And we have thought about this long and hard, and I've even asked transplant surgeons, well, what happens with innervation when you put the vessels back together? Do the nerves grow back together? Do they not grow back together? And the short answer is, we don't know. We need to know to fully appreciate this. What I can tell you, and what we know, innervated or not, and that's obviously the key point of the question, but innervated or not, if you put eyelids back in, and Sutherland has shown this as a transplant surgeon already over 20, 30 years ago in Denver, these new eyelids get attacked. It is a problem. So you can say this will remain a problem also for stem cell-derived eyelids. So we have to see. There's many things we need to still unravel, but these are questions right on the button.
Here's a question for Estefania. You know, the most essential part of our research program are recruiting the brightest minds that come and work here, the young researchers that build their careers. And what I've particularly loved about working with you are all the students that you train that tell me how excited they are for the things that you have taught them and the opportunity to work with you and work with Matthias. You have both an MD and a PhD. So you're a medical doctor and a researcher. Can you tell us a little bit about your path coming here, and then also at the same time address one of the audience questions about whether type 1 diabetes can be reversed through diet alone and not medication?
Yeah, so sure. I'm an MD. I'm actually a rheumatologist and a pediatric rheumatologist, and I'm also a PhD. I did my PhD in juvenile idiopathic arthritis, not in type 1 diabetes. But the reason why I'm investigating type 1 diabetes is because in autoimmune diseases, as Matthias already showed, there are many connections. Many of the problems that the immune system has in one disease are very similar to the problems that other diseases are having, even though the target organs are different. And genetically, interestingly, some of the diseases are actually quite close. And type 1 diabetes and juvenile idiopathic arthritis are relatively close. Some of the risk factors, genetically talking, are actually similar. And you can see aggregation of these autoimmune diseases in the same family, in the extended family, not immediate family. And yeah, sorry, and you like me to address one of the questions?
Yes, can it be addressed through diet alone and not medication? That's one. There's others in the Q&A. And I think people want to hear about your career path and what brought you here.
Well, what brought me here was actually La Jolla Institute for Immunology and Matthias. Matthias was the number one leader in the field, and La Jolla Institute for Immunology is, in my opinion, the best place in the world to study the immune system. So I wanted to do the postdoc in the best place with the best PI, and here I am. That was yeah. So fortunately, he gave me the opportunity. So I'm really grateful for that. And yeah, regarding the diet question, that's very interesting. There are a lot of studies going on nowadays, and not just in type 1 diabetes, in other autoimmune diseases. Actually, some of them are happening in UCSD, but some of our colleagues in rheumatology about the microbiome and its influence in the disease and its influence in the disease activity. But we have not yet fully figured out the field. It's a bit early days. I would say that having a good diet, it helps you to prevent some sort of inflammation in the body, and this is obviously helpful for the functioning of the immune system. But through the diet alone, once the disease starts, I think it's a bit late for that to correct it. Maybe to prevent like flares, but probably a bit late for correcting. There is a point of not return once an autoimmune disease starts for most of them.
Interesting that you bring up the point of no return. There's another audience question about would the therapies or the opportunities that Matthias, you've described, would they only work for recent onset patients, or would patients that were diagnosed years ago still be candidates for those kinds of therapies?
So that's a good question, and it's very clear. If the endogenous beta cells are all gone, then these therapies won't work anymore. But I would argue they are still very important because now we can make eyelids out of stem cells. So we will need them for these organ replacement strategies, and that's going to become a reality. So I look at this as an entire spectrum of continuum. And as I said, I think it would be blew out to assume you can, at least in the beginning, have completely immune evasive stealthed eyelids or a capsule that protects them so well you don't need to do at least a mild immunotherapy. And that's where the field is going to converge all together. So that's how that goes. They will work best in prevention, like many things, like for any disease. It's better to prevent type 2 diabetes than to have type 2 diabetes for any scenario. That's just how it goes. And that's the same in type 1 diabetes. But in type 1 diabetes, because it's a specific cell loss, the hope that you can do something about this by having an organ replacement is really a special icing on the cake, and probably now more proximal for sure than 10, 20 years ago. It's an exciting time.
Indeed. Another audience question. Could suppression of MHC1, the major histocompatibility complex, prevent type 1 diabetes? Any adverse side effects of that approach?
Well, if you do it everywhere in the body, the side effects will be catastrophic. You will make all kinds of mess with the immune system because the immune system can't recognize anything anymore. So you cannot just get rid of all your MHC molecules. If you do it in eyelids, most likely, yes. In the mice, it works like a charm. That has been done over 10, 20 years ago. The question becomes, how do you do this locally in humans? And the question also for eyelid replacement is suppression of upregulation of MHC on transplanted eyelids, which will become feasible. There's many companies working on that. Is that sufficient to make them not recognized? And I tend to think it will be a major factor. The only thing that will then still be active, if your eyelids are dying, they will still, whatever comes out of these eyelids from the death will still be presented by the immune system and reactivate inflammation. But I think the MHC is a major switch point, and we just published, it's not only class 1, but also class 2, Estefania's paper in Diabetologia this month with a cover, which we are extremely stoked about, to say the least.
Wonderful. Estefania, you brought up the microbiome, and this sparks some interest from the audience as well. Can you expand a little more on the role of microbiome in type 1 diabetes? And actually, it strikes me as that's one of the reasons why we all came here, is the ability of Jolla Institute to do that cross-pollination for microbiome researchers to influence those working on different diseases. And also, another audience member asked about the role of neuroimmunology in type 1 diabetes progression.
Okay, so answering the one about the microbiome. So first of all, full disclosure, I'm not an expert in microbiome. These are not the type of studies that we do at the Institute. And as I mentioned, so the microbiota, for those who are not really familiar with the term, refers not to the balance of the bacteria, like the good bacteria and the bad pathogens that we have in the intestines, and how this affects through their effect through the epithelia, and then the activation of the immune system locally, how this can spread or make autoimmune diseases worse. And there are several hypotheses out there about their capacity about how, when you have a disbalance for a long time, you can trigger an autoimmune disease, and how a disbalance in the microbiome can make worse an autoimmune disease, or can improve some of the symptoms of an autoimmune disease that already started. But as I mentioned, in type 1 diabetes, so once the disease starts, once there is an organized immune attack against the eyelids, modify it probably may help to have a bit of less of aggressive immune system, but difficulty at this point is going to revert it, especially when the beta cells are gone. I mean, are gone at this point. It's not really possible.
So the last question for Matthias: what research do we need to do, and what research do we need to support in order to prevent type 1 diabetes from beginning?
I would say two things if I had my choice. One is to continue with our crime scene investigations with the best people, the best equipment, and really to understand what's going on in the human disease. For me, pathology has always been a basis, especially human pathology, because you can build then models based on this and then understand more in detail. Secondly, I showed you the moonshot about neuroimmunology, and there was also the previous question. This is ready. It will require investment of an ecosystem that will consist of basic research, which one could do here, companies who translate the proper devices to modulate selectively and make this grow enough so it would become appealing then at some point for larger pharma and implementation in patients. I think the time is ready for this now. This we can do. We have all the tools. We should grow it. Probably not only for type 1, but also for other diseases. We need to understand it. And I think still it's really out of the box and quite appealing and cool, and would address things that we don't understand yet about the immunity, bringing you back to the symmetry of diseases. So these are sort of my wishful dreams and thinkings.
Well, it's very much the support of individual people and private families that lets us do things like that lets us capitalize on a new idea when the time is right. And it's private support that lets us recruit young researchers like Estefania when we find her, and we want to be able to support the projects that she's doing and the students that she's training. And it's also the private support that lets us buy the instruments that we need. So for example, in that game of Clue, the crime scene investigation, how do we know who did it, where, in what room, with what tool? We are looking to set Matthias up with a tissue analyzer that would provide that kind of information. But if you want to do the right investigation, you need the right tools.
Yes, that would be super nice.
We invite all of you to come to the Institute in person. Come and tour that microscopy core yourself and visit the Von Herrath lab and talk to us about how we can involve you in this very exciting research.
Yes, welcome anytime. It is very meaningful, actually, when we have visitors. Please, do not be shy.
And there's more information on how you can get involved and learn more on our closing slide. Thanks so much for your time and your attention.